So today we are going to talk about enzymes. Now you are learning about enzymes since you are in grade 6, right?
Yes.
Now you guys know that enzymes, they are called as biological catalysts. Now there are two words, biological and catalyst. A catalyst is a simple word from chemistry. It is something that speeds up the chemical reactions. Anything that speeds up a chemical reaction is called as a catalyst. But question is, why enzymes are called as biological catalysts? Why the word biological? Because they speed up the chemical reaction in living organisms. Right? So as they speed up the chemical reactions in the living organisms, that's why they are called as biological catalysts. Another thing which you guys already know, that all the enzymes, they are made up of proteins. They are made up of proteins. And remember one more thing, that these enzymes, they are all globular proteins. These are all globular proteins. Another very important point about the enzymes, that as they have a globular protein, but they have a site which is called as active site. Now, active site, it is a place in the enzyme where chemical reaction takes place. Or, if we be more specific, then we'll say that an active site is the site where substrates bind or attach to enzyme. So every enzyme has an active site, and at the active site, the substrates, they will bind. Now next question comes to your mind, that sir, what do you mean by the word substrate? Now the reactants of enzyme reaction are called as substrate. Reactants of enzyme catalyzed reactions, they are called as what? Substrates. Right? So when these substrates, they will bind to the active site, there can be two types of chemical reactions. Now on that basis, the enzymes are basically of two types. The enzymes, they can be either anabolic enzymes and they can be catabolic enzymes. Now next question is, that what is the meaning of catabolic and what is the meaning of anabolic? For that purpose, we need to understand one thing. We already know the word metabolism. Now what is metabolism? Who will tell me?
Overall chemical reaction inside the body.
Right? The chemical reactions taking place inside the cells or in our body, that is called metabolism. Now metabolism is basically divided into two types. It is divided into catabolism and anabolism. Now in our body, in our cells, the molecules are broken down or not? Now you eat something Like let's suppose that you ate bread. Bread is made up of starch. Starch is broken down inside your body or not? So larger molecules are broken down into smaller molecules. That comes under the category of catabolism. In catabolism, large molecules are broken down to smaller molecules, right? And in our body, building up of things also takes place. We have learned that in our body proteins are being made, right? So the making of protein is what? Smaller molecules are joined together. Amino acids are joined together to make large molecules, right? So when smaller molecules are joined to make the large molecules, that will be called what? Anabolism. So what is anabolism? Joining of small molecules to make large molecules. So if we come at the word anabolic enzymes, anabolism is building larger molecules. Now if you guys remember when you guys were young, you guys learned that there are two types of enzymes, builder enzymes and breaker enzymes, right? So the enzymes which are anabolic enzymes, we can call them as builder enzymes as well. Why? Because what they will do? They will join the small molecules to make the large molecules. They will join the small molecules to make the large molecules. That's why they are called the builder enzymes. Catabolic enzymes, they are performing catabolism, which is what? Breaking large molecules into smaller molecules. So simply these enzymes can be called what? Breaker enzymes. Right? Now, so there are two types of enzymes, builder enzymes and breaker enzymes. Next thing, if you come over here, we learn that all the enzymes, they are made up of what? Proteins. Right? And we also discussed that every enzyme has what? An active site. Now let's suppose that this over here, this is not the Pac-Man, this is enzyme. Now every enzyme has an active site. We just discussed it, right? So this over here, this is the, let's suppose, the active site of the enzyme. Yes?
Sir, could you give some examples of anabolic and catabolic enzymes?
I just gave the example. The enzymes that will be building up the proteins, they will be what? Anabolic enzymes. I gave you the example of the catabolic enzyme, the ones which are digesting the food which you ate. Starch, what is the name of the enzyme that breaks down starch? Amylase. So that is what? A catabolic enzyme. Alright? So I already gave the examples. Please pay attention. Next thing. If you see, I made another bend, you see dent over here, other than the active site. Now, the enzymes, they have another site other than active site. This is called as allosteric site. What is it called? Allosteric site. Now, with this allosteric site, a coenzyme comes and binds. A coenzyme or a cofactor comes and binds with it. Now question is, sir, what do you mean by a coenzyme or a cofactor? Now let me tell you something, that this enzyme which I drew, this is the protein part of the enzyme. This is all protein. Now this whole just protein part of the enzyme, this is simply called as apoenzyme. Alone apoenzyme is nothing. It can't do its work. Now, you are a student, right? You come to school. If you don't bring your books, you don't bring your markers, your pencils, whatever, right? Will you be able to learn anything? No. I'm a teacher. I come to class without my weapons. Will I be able to teach you? No, right? Same. This is enzyme, but this is the protein part. But it will become active when a coenzyme or a cofactor binds to the site which is called allosteric site. Which is called what? Allosteric site, right? Now, very important thing that a coenzyme or a cofactor, when it binds with the apoenzyme, it is called as holoenzyme. It's not holo, H-O-L-L-O-W, that's not. This word holo, it means complete enzyme. It means complete enzyme. Now let me show you in the form of an example, in the form of a diagram. Again, let's suppose that this is what? This is the enzyme. This is its active site. And this is its allosteric site, right? Let me label that. This is the active site. And this over here, this is the allosteric site. And let's suppose that this over here, this is what? This is the cofactor, right? Now, when both of them, they will bind together like this. Now what has happened now? This cofactor has bind to the allosteric site. Now this is what? This is the holoenzyme. Understood? Now this enzyme is active. Now this enzyme can work. Understand? Next question which should have come in your mind, that what is the difference between a cofactor and coenzyme? Now, if you guys remember, you guys have learned in O levels, in the nutrition chapter, that we require minerals in small quantity in our diet. Remember that or not? You guys have also learned that we require vitamins in our body, in our diet, in small quantity. Right? Now, where these minerals and vitamins are required? Now, some of the minerals like calcium, magnesium, iron, they are the inorganic ions, they are the cofactors. Inorganic ions, what are they called? Cofactors. Right? While if the cofactor, which you see, or the part or the thing which activates the enzyme, is an organic molecule. For example, vitamins. For example, vitamins, right? So these vitamins which you take in your diet or some other organic compounds, when they act as cofactor, that will be called as coenzyme. Understand? So what is their job? They activate the enzyme. They help in starting the chemical reactions. Understood? Another very important point about the enzymes is that enzymes are required in very small quantity. The question is, why enzymes are required in a small quantity? Because enzymes can be reused again and again. Once an enzyme performs either anabolic or catabolic reaction, or we simply say metabolic reaction, at the end it can be reused. How can I prove it? Now, let's suppose that over here, this is what? This is an enzyme, right? Let's suppose that this is the active site of the enzyme. Let's suppose that these are two substrates . As there are two substrates, it simply means that this is what? A builder enzyme, an anabolic enzyme, right? What will happen? That these substrates, they will bind to the active site. Let's see that these bind to the active site. Now, when the substrates they bind to the active site, this union of enzyme and substrate, this is called as enzyme substrate complex. What is it called? Enzyme substrate complex. Now, let me tell you one thing, that this enzyme substrate complex is formed for a very short period of time, for the fraction of a second. Right away, what happens? That this enzyme, it becomes separate, and the product also becomes separate. So over here, this is what? This is the product, and this is what? Enzyme. Now, what was there? In the beginning, there were two small separate molecules, right? Which were substrates. They bind to the active site. In the active site, enzyme helped them to join them together. And what happened? That one larger molecule came out. And enzyme became free. And you guys can see that the active site is free again. The active site is free again. As the active site is free again, so it can be reused in the chemical reactions. So enzyme can be reused in the chemical reactions because the active site became free. And there is no change in the enzyme. Now, if you guys remember, you guys have learned in catalysts as well, in chemistry. It is something which lowers the activation energy. Remember that or not? Speeds up the chemical reaction and is not used up in the chemical reaction. So from this point, we deduce another point in the enzymes properties, that enzymes do not get used up in chemical reactions and can be reused So as the enzymes can be reused, that is why small quantity of the enzymes is required. Understand? So that is why, you see, in our cells, thousands of chemical reactions are taking at the same time. As thousands of chemical reactions are taking place at the same time, if the enzymes, you see, our cell is losing enzymes every other minute, what will happen? We will not have enough energy to build new enzymes, right? So enzymes are reused again and again, and that's why they are required in small quantity. Another thing which we will learn in detail inshallah later. The thing is that which you guys have learned before as well, that enzymes are affected by change in pH. You remember this or not? Increase or decrease in pH can denature the enzymes. Now, how increase or decrease in the pH can denature the enzymes, we will discuss in detail. But right now, just let me give you a little idea. Let's suppose that there is an enzyme that works at 7 pH. Most of the enzymes work at 7 pH, let me tell you that. There are only few enzymes that either work in the acidic pH or in the alkaline pH. Now, an enzyme that works at 7 pH, if let's suppose we change the pH to acidic, then what will happen? Number of hydrogen ions will increase, which will change the polarity of the solution. Now, if you guys remember, what did we learn? That tertiary structure of the polypeptide is maintained by hydrophobic hydrophilic interactions. So the tertiary structure will be destroyed. And when the tertiary structure will be destroyed, so eventually what will happen? Quaternary structure, the globular structure will also be destroyed. So once the globular structure is destroyed, what do we say? Active site is damaged. What do we say? That the active site is damaged. So once the active site is destroyed, we say that enzyme is denatured. Understood?
Yes.
Another very important point, which I will write somewhere over here, that let's write over here that high temperature and which is above 40 degrees, and you see, and you guys remember, at 40 degrees enzymes they start to get denatured, right? And at 70 degrees enzymes they completely denature. Now, remembering the structure of proteins, can you tell me a reason that why enzymes they will become denatured at higher temperature?
The hydrogen bonds will break.
Excellent answer. Well done. The hydrogen bonds they will break. The secondary structure will be disrupted. Even the tertiary structure, because remember, secondary structure, tertiary structure, it is maintained by hydrogen bonds, ionic bonds, disulfide bridges in the quaternary structure, right? So hydrogen bonds they will be broken down. Will the globular structure of the enzyme be maintained? No. So what we'll say, that enzyme's active site is denatured. That substrate will not be able to fit inside the active site. When the substrate will not be able to fit into the active site, eventually what will happen? Enzyme will denature. Understand? So these are some of the general properties of the enzymes. Now, next lecture inshallah, we will talk about how enzymes work. Now, no one has seen the enzymes. How do they work? They're molecules, right? But with the help of experiments, we can prove that how the enzymes work. Or scientists, they can give assumptions. They can give their hypotheses that how do the enzymes work. Now, on the basis of experiments and on the basis of hypotheses of the scientists, we have two models which are represented that how do the enzymes work. One is called as lock and key model. You have learned about lock and key model in O levels. Second is called induced fit hypothesis or induced fit model. Now, induced fit model says that the shape of the active site is not exactly like a substrate. Remember in the lock and key model what we have learned, as there is a specific key for a lock, there is a specific substrate for its enzyme's active site, right? But in the induced fit, I will draw the diagrams and explain. You don't worry, inshallah. So what does it say? It's not an exact fit. But when the substrate binds to the active site, it becomes the exact fit. Now let me give you the example for that. Then we'll stop today's lecture. If I buy gloves, if I wear those gloves, they will become the shape of my hand. They are already shape of a hand, but not exact shape of my hand. Right? They have 5 fingers and you see a palm area. They are the shape of the hand, but not exactly my hand shape. If I will wear them, they will become exactly my hand shape. So this idea is given in the induced fit, that when before the active site matches, not the exactly, matches the shape of the substrate, but it becomes an exact fit when the substrate binds to it. Right? That is called as induced fit model. Alright?